Good afternoon, everybody, and thanks for coming. I'm going to talk to you about a particular investigation within a project, and you've got some information there about the group that's involved in this. Basically, it's a collaboration between a systems engineering group at the University of Reading who are doing some development work for us in haptics, and I'll tell you a little bit about haptics because that's obviously something that we need to make sure we understand something about here. And the Education Department at King's College London, where I work. And we also have close collaboration with a couple of schools, and one of the school teachers, Ros, has been heavily involved in the design and work on this project. Okay, so the title includes haptics, which I'll explain, and we're also looking specifically at understanding of difficult concepts, and this particular investigation was in science learning and specifically in biology learning, but if you're not a biologist, I will try and explain the key issues there that we're attempting to deal with. But overall, the project is not just focusing in science and biology, it's looking at, this is one example of maybe what might be possible in the future. Okay, so I'm planning to, well, the aims of the project, first of all, we needed to design a haptic-enabled virtual reality environment. And I'm assuming that we're in this session because it's virtual reality and it's haptics and it's simulations. We're using simulations. We're not using games as such, but obviously there's a link between those. So that students could explore these difficult concepts in this multi-sensory environment, and students could collaborate. So we had the aim of producing something that enabled all that, which in itself is quite complex. And then we wanted to examine if there are any particular effects of the haptics, and the haptics is virtual touch, and I'll tell you a bit more about how we're actually implementing that in a minute. And so we were trying to measure the effects, but also to look at students' perspectives on that. So I'm going to tell you a bit about the background and the theory and why we think there might be some merit in doing this. So we're looking at the challenges of understanding something about cell membrane structure. We're working with students in their early years in secondary school. And so I'm going to say a bit about the design of the virtual reality environment, a bit about the research design, and something about the findings that we have so far and our conclusions. So first of all, what is haptics? Well, it enables you to touch things, and the particular interface that we've developed enables you to touch things with a thumb and a finger within a virtual reality environment. So people doing this will be wearing an Oculus Rift, so they're in this virtual reality environment, and they can touch things, move them around like this in that environment. Which is a step forward beyond what we had before, where we were just able to point at things. And it's definitely a step forward over just being able to use a mouse to touch things and move them around. We can, with this haptics, we can perceive texture and forces directly. In particular, we're focusing on forces in the particular investigation we were doing. So it's better than using a mouse, and also you get this extra sensory, this extra sense, not extra sensory, but extra sense. So there are already known benefits of multisensory learning compared with unisensory, particularly with students who have special needs, but with all students. We think this is likely to be engaging and motivating because it's more realistic than the kinds of interaction that you can do without having the touch enabled. We also think that it might support visualization in some areas of science, because obviously there are quite a few things that you do in science, and especially biology, that you can't interact directly with them. You can't interact directly with cells because they're too small, and membrane structures are incredibly small. So we're looking at this kind of nanoscale, particularly where you can't interact unless you have a simulation in a virtual reality environment or some sort of virtual environment. There is a potential theoretical foundation for this, because there is the dual coding theory, which proposes that if you're getting impulses from different sensory stimuli, then they might act together and give you a better understanding than one sense on its own. Cell membranes, we chose these. We started off with looking at the whole cell, but I'm focusing here on cell membranes because this is the particular study we've just done. And we know that cell membranes are incredibly important in biology. They're also very difficult to make sense of. There are some difficult concepts, and we know there are quite a few misconceptions. And we also know that there are forces at work across the cell membrane which you can feel, and you can push things backwards and forwards across the cell membrane, and you can feel those forces in a virtual reality environment. So that's why we chose this particular topic. Those are some of the difficulties that students have in understanding. The plasma membrane is the same as the cell membrane. It's this membrane around the outside of a cell. But there are obviously lots of other membranes in cells as well as the outside membrane. So we were focusing particularly on the membrane that surrounds the cell. I won't read through all of those because you can read, and I haven't got a lot more to say about them than what's up there. So you can just see that there are quite a lot of difficulties in understanding cell membranes. And there are some very specific misconceptions which we're trying to get at. We were trying to get at it in this particular study, and we're going on doing some further studies to try and get at them a little bit more. So what we want students to understand is that some molecules are able to diffuse freely across the plasma membrane, whereas others cannot, because obviously the cell membrane, the plasma membrane, controls what enters and exits the cell. And that's really important for all sorts of processes that go on inside cells. Students tend to learn about diffusion in physics as well as in biology, and there they learn about the way molecules move through a liquid. They don't necessarily bring that understanding into biology, and even if they do, then there is this barrier that stops some molecules and not others for various different reasons which they won't necessarily understand at the age we're looking at. But they will hopefully get this idea that there is simple diffusion going on for some molecules, and for other molecules they need a more sophisticated method of getting across the cell membrane, which is built into the structure of the membrane by specific membrane transport proteins which enable molecules to move across the membrane. And they can find out a little bit about how that works through this simulation. So what we had to do was to think about how to design the virtual reality environment, because as well as designing the haptic interaction, we had to, the engineers at Reading had to write the software to actually enable this to work. We had some support from a company that developed 3D models as well, but obviously that then had to be further modelled. And we had to work out, well, what level of complexity are we going to use? Because cell membranes are incredibly complicated. We couldn't just take a picture of a cell membrane, a photo of a cell membrane, and implement that. It's too complicated for that. So we needed to decide on what level of detail to represent. And we also had to think about how to model the relative size and scale of the structures. We know from what I presented on the previous slide that scale is something that students find very difficult to understand, but it's actually not that easy to present either. And in textbooks, it's always very badly represented. And even in this virtual reality environment, we just still had difficulty in working out how to cope with the scale issues. And then we had to think about how to model the haptic forces so that students would be able to feel them and manipulate them. So there were quite a lot of technical challenges as well as the educational ones. So this is what the cell membrane model actually looks like. So there you can see a cell membrane that doesn't look all that much like a cell membrane actually. But here you have what we call the phospholipid bilayer. It's made up of a bilayer of phospholipid molecules. And it doesn't really look like a completely smooth, flat wafer. And it does move around, and we haven't been able to simulate that terribly brilliantly, but at least you can see that it's a barrier. And you can see that there's, it's not solid. There are bits here that are different, so it's not a completely uniform structure. What is a little bit more realistic, but still not all that realistic, is that these particles are able to move around, and they move around by Brownian motion and also under the forces of diffusion, under the diffusion gradient. So what we've got here is, this is the outside of the cell, and this is the cell membrane. And over here we have the inside of the cell, which is called the cytoplasm. And these objects here are molecules. So we've got, these represent, in this particular screenshot, these represent oxygen molecules, and they are two red blobs because that's the typical way that oxygen atoms are represented by red spheres, and we're representing O2, so they look like that. So the students get to make a bit of sense of them because they possibly know that oxygen is O2, and they might have seen them represented like that because they're often drawn like that, and that's a standard representation. And these are carbon dioxide. We also implemented glucose molecules, which are much bigger than that, and they're not shown in this particular screenshot. And we have some potassium and sodium ions as well. And the sizes of the molecules are roughly accurate. But of course they don't all look the same size there, because you're seeing this, you're seeing a picture of a 3D representation. So depending on how close you are to the actual molecule, they're going to appear different sizes. And as the students move around, they will be able to make sense of that. The things that project through the membrane here are membrane proteins, and some of those are glucose transporters. So if the students are able to grab hold of a glucose molecule and get it near to a glucose transporter, it will whiz through the membrane, which they find quite exciting. With these, they can pick these up. A label appears to say what it is, to remind them what kind of molecule they're picking up. And as they push that towards the membrane, it will go through, and how easy it will go through depends on the relative concentration of the oxygen molecules in that model. And they can add extra oxygen to the model. And the same with carbon dioxide. So that's the basic model. And we had them working from a worksheet. And we needed to design this in such a way that we didn't interact with the students while they were doing this at all. Obviously, in a normal classroom situation, the teacher would interact, but we wanted to set this up as an experiment so we could see whether it made any difference, whether they were able to feel the molecules or not. So that is to remind them of what they've seen in the textbook, which is basically going to be mostly a two-dimensional picture like that, and then get them to make sense of the model, which is just this little bit of the membrane there, but shown in 3 dimensions. So hopefully that makes sense to you. If you're not a biologist, you get a rough idea as to what it is we're trying to achieve with that. So that's what it looks like when they're using the system. So we experimented in previous studies with various ways of having these students interacting. So what we've got here, this person is the pilot, and they happen to be left-handed, it works with iron. They are interacting with the haptic device, they have the Oculus Rift there, and this person is the co-pilot, and you'll see that she is writing on a worksheet. So she has a paper-based worksheet and is able to read out the questions and the pilot is able to move around and interact with the system. The co-pilot can see the same sort of representation that we just saw a picture of. So she sees that in what is nearly, well, it's a sort of three-dimensional representation. She can get a rough idea of that, so she knows what the pilot is, roughly what the pilot is seeing. And we found that this works quite well because the co-pilot is able to explain to the pilot what she's supposed to be doing, and she keeps her in touch with the real world as well. That's what the students tended to say, that they felt they were grounded in the real world a little bit, whilst at the same time being quite immersed in this environment when they were interacting. So that's the way we set up the...
3 minutes.
3 minutes left. Okay, I'm going to have 3 minutes. Right, I shall whiz through this a bit faster. Okay. So those were the key concepts that we were focusing on, and I've actually mostly explained those already. Yeah, I've explained all those. So that's what we were trying to get them to learn. There's the research design. We had 2 scores. We got 64 students. The experimental group had the haptic-enabled interface. Now, in fact, all of them, the way they were interacting looked exactly the same as what you just saw, except that we were able to, in the software, to turn off the haptics so that in the control group, they couldn't actually feel. They could still move things around, but they couldn't feel. So we did pre- and post-test, which had some true and false questions. It had some other questions as well, but we haven't finished analysing those yet, so we're only presenting that evidence. And we also took field notes and video recorded, and we interviewed the students to get their perspectives. We don't need to see that again. So those were the results from the pre and post tests. So basically they did learn, and there was a significant difference between the pre and post test results. But there wasn't a significant difference between the haptic and the non-haptic condition. So what we're doing now is to try and think, well, why was that? And it could just be that they really don't need to be able to feel. Just being able to interact is enough. It's a fairly authentic kind of interaction. It's quite dynamic and interesting. The students' perspectives, we've analysed the interview findings. They found the system easy to use, so that was fine. A few of them found difficulty in grasping the objects, but that didn't seem to be related to whether or not they could feel them, actually. It was possibly related to how good they were at grasping things, and that's something we're looking into. They all liked working in pairs, particularly in this environment. This setup worked well. And they all thought that the way we had the system with the pilot and the co-pilot supported them in collaborative learning, which they liked in this particular setup. So that worked. Thinking particularly about the importance of haptics, the ones that were actually being able to feel, they believed that that helped them to gain a better understanding. They certainly liked being able to feel, and it helped them to gain a better understanding compared with the way they were normally taught this subject. But the ones that were in the non-haptic condition were also quite positive about this. Well, they were very positive about the system, and they liked being able to grasp the objects and move them around. So the question is, do we need this haptic feel or just being able to interact? And some of them who were not able to feel, they were saying that, well, actually you can't feel, but you can sort of imagine that you're feeling because you can see it. And this is actually what surgeons do, because most surgeons, when they're using robotic interfaces, they don't have haptic enabled interfaces. They are just able to perform operations by looking at what they're doing. So maybe that's enough. We can't be sure yet, but it's a possibility. So that's what we've concluded. It was successful. We have got a virtual environment in which students are able to learn, they are engaged, and it does support collaborative learning, and we do like this setup with the pilot and co-pilot. The haptic feedback, it was certainly engaging and probably useful as far as the students were concerned. That was their perspective, but we haven't managed to show that it actually improves their learning. So thank you very much. That's the end.
Thank you, Mary.